Hi everyone. So we'll get started just so that we've got enough time at the end for you to quiz Pip on her work, and even if you've got time to talk about education at the university as well. So today we've got Professor Philippa Pattison, who was appointed Deputy Vice-Chancellor for Education at the university in June 2014, so about a year ago. Within that role, she's responsible for the university's strategy and vision for both teaching and learning, but also thinking about the students' educational experience. By doing that, she is overseeing the institution-wide development of student learning, but also our approach to curriculum renewal, think about new techniques in pedagogy, learning, and teaching analytics. But today, we're actually moving away from that and thinking about more of an infection focus. So Pip obtained her BSc Honours and PhD in Psychology from the University of Melbourne, and then she was with the University of Melbourne more or less from then onwards until she came to Sydney.
Thank you for not counting.
I didn't put dates in there. She started off as a lecturer in psychology, rose through the ranks to professor, and then took on various leadership roles, including head of the School of Behavioural Science, deputy vice president, and ultimately president of the academic board. Pip became Pro-Vice-Chancellor for Learning and Teaching and then also Deputy Vice-Chancellor Academic in 2011. The primary focus of Pip's research is the development and application of mathematical and statistical models for social networks and also network processes. Recent applications of this have been in the transmission of infectious diseases, which she'll talk about today, as well as the evolution of the biotechnology industry in Australia and community recovery following the 2009 Victorian bushfires. So, without further ado, I'll hand over to Professor Pattison for today's talk. Thank you.
Thank you, Grant, and thank you. It's actually a real pleasure to be here and to be talking about my former life as much as my current one. I might break into a talk on education if I'm not careful, but I'm actually thrilled to be talking about networks. I'm going to talk about joint work with many colleagues, some from the University of Melbourne, but also from other parts of the world, and particularly with colleagues from the Burnish Institute in Melbourne, who have the disease expertise in hepatitis C virus that I'll be talking about, and who also initiated the study that I'm going to be analysing today. What I want to do is talk about 3 steps in a modelling process, each of which I think is very important to understanding disease transmission at the population level. First of all, as I'll mention briefly, but I think is evident to many, is that the structure and dynamics of contact networks can be really important to disease transmission, but we don't actually have very good models always of what those contact networks are like. And so what I want to talk about in part today is the process of actually empirically discovering and estimating the structure and form of those networks. But of course also the disease transmission process itself at a personal or interpersonal level is important. And I'll talk about building a model for that. And then we can put those 2 components together and have the model for disease transmission actually operate across a network that's been empirically estimated to give us a much better model of the dynamics of disease transmission that we can then use for a variety of different purposes, but including that of predicting the likely impact of different treatment strategies that we might anticipate. So just to sort of start with the, I guess, obvious point that transmission of hepatitis C virus is very much a byproduct of what is an otherwise completely unrelated social interaction. And the particular social interaction that we're going to be looking at is that of injecting together using intravenous drugs. And in many ways, it's a completely sort of separate question as to how those networks arise and how they're structured to that of the disease transmission itself, although in many cases that we can imagine, these 2 issues do come to be coupled. But to begin with, I think we'll approach that by considering the social process itself. And as you've heard, I'm a social scientist rather than a medical one. And the second point is the one I've also just made, that the structure of those networks is vitally important to the spread of the disease. And I think we saw in the recent Ebola outbreak the commentary around the changing rates of infection were to do as much with the changing nature of social interaction as with other things. So the social part of this is quite important. Now, if we're interested in the nature, the structure of networks of people who inject together, we need to actually find some empirical means of discovering what those are like. And of course, a complete census of contacts in the population is infeasible. And if we use our more customary approaches to gathering data using some kind of sampling strategy, if we simply sample individuals from the general population, we'll actually discover relatively few intravenous drug users. If we start with intravenous drug users, that is, if we can find them, and we just sample randomly from them, we'll also discover relatively few connections or contacts between them, because these networks tend to be quite sparse. And so what people have begun to do in this kind of work with sparse networks and hard-to-find populations is develop what are called snowball or contact tracing sampling designs, where you find some members of the population of interest and then you trace their contacts and then their contacts of those people and so on, and you actually uncover fragments of the network of interest rather than the whole network, but at least you discover some features of it. Now, of course, everyone who's had an interest in statistics knows that you immediately have to be concerned about the possible biases that this might create in the sample. And so we need to actually build models for the contact networks that are aware of and that can accommodate any potential sources of bias. But the more general problem, of which this is an example, that of adaptive sampling, is the subject of very active work in statistics at the moment, and is actually a very useful approach for many problems. So to begin with the data that we're going to analyse, this is from the study that Margaret Hellard and others at the Burnett initiated some years ago now. And they started with, if you can see the nodes are coloured, the yellow nodes, these were actually the initial participants in a study. These were intravenous drug users that the research team identified in 3 locations in Melbourne. And each participant was asked to name the people with whom they inject drugs, indeed if there were any, and you can see a number of the yellow nodes have no connections at all. They injected alone. Some just inject with a partner, and those are the pairs which are just a yellow node and a blue node together. But in every case, the blue nodes were also asked with whom they injected, and that led to the identification of the pink nodes. At that point, the money for data collection ran out, and so that's the sample we have to work with. And you can see that even though we start with a relatively small number of nodes from these locations, the parts of the network are beginning to join up, and one of the questions we want to know is, how big is this network? Does it, go on indefinitely, or is it of a smaller size? And what does the shape of that network look like if we're interested in HCV transmission? So the first question we ask is how we can use data of that form, fragments of a contact network that have been gathered by this contact tracing design, to model the whole network. The answer is we can, and it's a slightly sort of a prolonged story to get there. Please bear with me. I was just saying to Grant before, I've cut this presentation down so that you only see one equation, but if anyone wants to see more, please let me know. I have more. So let me just start by saying something about how we think about modelling a network. We regard the nodes of the network as fixed and treat as variables the potential contacts between the nodes. So if you're thinking about statistics, it's the lines in this diagram that are the variables, not the points on the diagram. But of course, because edges or connections or contacts in this network often share a particular node, we can't regard these lines as independent of one another. So we're actually building a model for a collection of variables, each of which corresponds to a potential tie, and those variables are potentially interdependent. And so the process of model building needs to deal with that potential interdependence, primarily by making some assumptions about its form and then testing out whether those assumptions are reasonable ones. And we assume in the process that some effects that we might be interested in, for example, the tendency for the partners of a particular node in the network to themselves be tied, they might be common across the network. And what we do is we build a model that we can estimate from a single observation of the whole network. Of course, the problem we're about to get to is that of building that model from just a fragment of the network, but that's a slightly harder problem. So, a critical issue in building the models is how we think about the relationships or dependencies between these tie variables. And we essentially begin with the assumption that if tie variables are very far away from each other in the network, they're unlikely to be dependent, but dependencies are much more likely the closer they become. But how close do they need to be? And this has been a 15-year journey to discover the answer to that question, and I think we've got some reasonably good answers, not that we don't necessarily know everything. But in thinking about how close they might need to be, it's useful to have a look at the literature on propositions about the kinds of mechanisms that lead to structure in networks. And this slide simply gathers together a number of the theoretical claims about structuring mechanisms in networks that have particular implications for network structures. So, for example, the very first one, Cartwright and Harari, was a claim about, you know, the friends of my friends are friends, the enemies of my enemies are friends, and the friends of my enemies and enemies are all that sort of thing. And that then makes a very clear prediction about the structure of the networks that can be formed of friendship and non-friendship ties. But there's a whole range of those. They're just there to illustrate that much of the theoretical thinking about the structure of networks has honed in on quite small structural features, because most of the mechanisms are local in the sense that I mentioned before, that dependencies between tie variables are likely to be strong where they're somehow close in the network. They don't have to be adjacent, and a good example of that is in this case. This was a study by Peter Behrman and others who were looking at the structure of what they called romantic or sexual networks. I think romantic was there to satisfy the sensibilities of adolescents' parents, but they did the Ad Health study, a big multi-site study of adolescent health in the US. And they were interested in what kinds of mechanisms might drive the kinds of networks that you see when you actually put together all the information of who is whose boyfriend or girlfriend. And the mechanism that they honed in on as being particularly effective in this case was one in which you tended not to see what you could call partner swapping. That is, if Bob and Carol had a relationship at one point in time, as had Alice and Ted, and if Carol and Ted then get together, typically Bob and Alice don't. And there was a very bad movie with the name, something like that. Most of you would be too young to know it. But that mechanism would predict a network structure in which there are relatively few of these four-cycle structures. And of course, apart from those sort of structural mechanisms themselves, There are other sources or other characteristics of individuals and their settings which predict the formation of contact ties. Certain sorts of personal characteristics and their similarity, or in some cases their difference, are predictive of the formation of ties. Having similar interests and ending up in similar places is predictive of network ties. And then there are all sorts of geographical, organisational, socio-cultural kind of framings of our personal lives that have an impact on the networks that we create. And indeed, we can even see interdependence with, say, a network like injecting together that will obviously partly depend on things like friendship networks and kinship networks and all sorts of other things. So this is the one equation, and I'll just quickly talk to it, but if this makes no sense, don't worry too much. When we think about modelling a network, we're thinking about modelling the ties, and so that the variables in this system we can represent as a connection between a node I and a node J. We call that Y. It's a binary variable, it's on or off, at least in this simple account. And of course, on any one occasion, if we look at that network, some of the ties will be on and some of the ties will be off, and that's a realisation of that network at that moment in time. And what we do is we build a probability for that network, so the probability of seeing any particular pattern of on and off values for the ties, we model as a function of certain characteristics of the network. And this is a consequence of the interdependence of the ties, that we have this kind of self-referential form to the model. So the model becomes an expression of probability in terms of particular parameters and network statistics that describe the tendency for particular sorts of structural features to happen. So, for example, in the case of the romantic network that I just showed you, if one of the statistics is the number of 4 cycles in the model, and if the prediction is that we see very few 4 cycles, we would expect to see a very negative parameter for the number of those 4 cycles. And in that way, we can conceive of a model in which there are certain tendencies for certain structural features to occur more likely than often or less likely than often. And there's some complicated statistics around this in the sense that in order to ensure that this is indeed a probability distribution, the denominator of this expression, kappa, which is the sum of the probabilities of all possible realisations of the network, is generally incomputable because of the number of possibilities that need to be worked out, and that means you need to engage in some clever computational statistics to avoid having to ever calculate that value. No more maths. So I'm going to show you 2 particular versions of this model, one of which is very well known, and this is the first one. In a limiting case where there are no structural tendencies in the network, all of the ties occur completely at random. And this is a very well studied model, originally developed by Paul Erdos and his colleague Renyi, and has given rise to the sort of subfield of random graph theory. So in this model, every tie is equally likely, and every possible realisation of the graph is equally likely. And in that simple situation, you can work out lots of things about how that network model works. And while it's a beautiful model to work with mathematically, it's very rare to see any observed network that has the characteristics that are predicted by this model. So we've been messing around with modelling networks for a while, and we found this is a useful one. And it very much picks up on this idea that Behrman and colleagues were thinking about when they were thinking about romantic networks, but it's much more general than that, because here we entertain the possibility that 2 ties in a network might be conditionally dependent, but only in the case where, if they came into being, they would create a 4-cycle or something shorter. So it's that sense, that's what we mean by being close in the network. So if we think of the 2 dotted lines as random variables, we're thinking of those as being conditionally dependent when the 2 red ties are actually in place. But if one of those was to disappear, they would no longer be dependent. And the prediction from this model is that the sufficient statistics, or the network statistics that we need to worry about in modelling this network, are things like the structure on the left, which is 2 nodes having a number of shared partners, or 2 connected nodes having a number of shared partners, or there being even more densely connected substructures of some size. And these can have many nodes or relatively few nodes, but they all have this property that every pair of edges lies on a short cycle in the network that describes the sufficient statistic. And we really think about this as a network that arises from some realisation-dependent closure process or anti-closure process. In the case of the romantic network, it was an anti-closure process, but in many social situations, there'll be a closure process at work. And this actually leads to a model that reproduces the characteristics of observed networks rather well in many cases, and is the only known model to do it. But we've been thinking about network models, and we've developed a hierarchy of possibilities driven by some very basic graph theoretical logic. And the model in question is the one in the circle there. It represents a closure process, but there are more open kinds of network processes we can envisage if we need to, if this proves unsatisfactory. And there are some network processes that are much more about even more cohesive structures. So there's actually a whole family of models that we can work with if we need to. And the statistics or network effects that are associated with this model are mainly these 4. The propensity for an edge to occur, so that controls how sparse or dense the network is. The extent to which the degrees of each node, that's the number of contacts of each node in the network, the degree to which that's a very diverse or dispersed distribution across the network or more homogeneous. The propensity for a pair of nodes to share partners, and we build this in a way that there's a progressive discounting for additional partners. And perhaps most importantly, the last one, the propensity for nodes that share many partners to themselves be connected, and that's very much a closure process. So, with that model in mind, we now just have to solve the problem of how to estimate a model of this form when we don't actually have the whole network. We've only got the fragments that I showed you before. And the thinking is basically this. When we're actually observing a network, and imagine that's the whole thing, if we randomly sample some nodes, say the yellow ones in the middle, and we trace their connections to the green nodes, and then we take the green nodes and we trace their connections to the red nodes, we take the red nodes and we trace their connections to the blue ones, we eventually see all of the network as long as it's connected. Of course, it may not be. But the insight that actually allows us to solve the problem of working with the fragments is that with the assumption of nodes only being conditionally dependent if they lie on a potential 4-cycle, if we start thinking about modelling the ties amongst the green and yellow nodes, we can quickly see that with that assumption of conditional dependence, that there may be a dependence on ties amongst the green and yellow nodes, on ties between green and red nodes, but they'll be conditionally independent of ties with blue nodes, or yes, any tie involving a blue node. So that actually allows us to condition on that part of the network that sits outside the red nodes, but the model is still the same, so that we can actually use the data amongst the green and yellow nodes. We can regard as temporarily fixed the ties between the green and red nodes, and we can disregard the ties between the red and blue nodes and anything that lies beyond. And that's basically the mechanism that allows us to estimate the model from the fragments. And so if we go back to our network, and we actually do that, we end up with a model for the network which allows us to say something about these effects in the network. We see that the edge parameter is very small and negative. It's a very sparse network. I've added in a term for isolates just to account for the fact that there's quite a high tendency for those isolated yellow nodes to occur, but we could have disregarded those because they're really outside the connected system. There's no particular positive or negative tendency to high dispersion in degrees, so the degrees are all, somewhat similar. There's a very strong tendency of this clustering effect, so it's very likely that if you share injecting partners, you'll inject together. But it's quite unlikely that if you share injecting partners, you won't be injecting together. So this is really very much a story of social clustering. There's also some dependence on individual characteristics. Gender is not important here, but being a user at the same level of frequency predicts a stronger contact. Being in the same age bracket, below or above 25, predicts a tie, and of course being in a similar location also increases the prediction of a tie. Now that sort of tells us something about the social process that gives rise to injecting together, but we also want to use that to actually build a model for the network itself. But first of all, we need to figure out how big the network is, because we actually don't know how large the network component from which that sample was drawn is. But fortunately, there's enough information in the structure of the network that we've already identified to actually help us do that. And so what we did was take the parameters of the model that we'd estimated and actually kind of imagine networks of different sizes with those parameters, and we just simply simulated them and looked to see whether or not they predicted various characteristics of the network that we were using to estimate a whole lot of characteristics that we hadn't used to estimate the network, but we could work out from the network. And interestingly, and this is presented in the form here, that converges on a network size of approximately 524. So values much lower than that were inconsistent with various features that we saw. Values much above that were inconsistent with some of the features that we saw. So that really kind of narrowed down the range of network sizes that were consistent with the model. And we can then take that estimate to simulate the network as a whole. Simulation is not completely straightforward, but you can use a well-known technique of Markov chain Monte Carlo sampling to create a process to sort of sample from this the model, possible realisations from the model. And there's an example of one such instantiation or realisation of the network. So that's the first part done. We can model the network, that's great, but we also need a model for transmission at the more personal level. This is not my area of expertise, but David Rolls, who led this part of the study, worked very closely with Emma McBride and Margaret on the data sources and the infection process that might be consistent with what we know and what we were able to observe for these particular individuals. And I should say blood was taken in every case, and we know quite a lot about the virus that each of the individuals involved in the study who had an infection was infected with. And I should have said at the start that we originally did some work to make sure that there was some correlation between the injecting network as we measured it through the contact tracing process and the phylogenetic distance between the viruses themselves, if that makes any sense to you. I'm not sure I'm using the right words, but it just said there was a biological and social correlation. But anyway, David modelled the process and estimated a lot of the parameters of this process and basically gives rise to an agent-based model for, you know, given that one person is injected, what's the probability of the partner being injected? The model had 2 bits to it. There was the first bit, I should go back. Whoops, wrong way. This first bit is the probability of a transmission, and the second part is the duration of that infection once it's occurred. And so the next step was to take that transmission model, combine it with the contact model, and actually model transmission at the whole of network level. And to do that, we've got a model of the network, so we can actually, it's a probability model, we can draw many realisations of that model, so many realisations of the network, and in each case we can simulate transmission and compute various measures of outcome. For example, what's the proportion of individuals with an infection at a particular point in time? So we did that. And then we did that because we were very interested in trying to use that model to understand whether there would be different population level outcomes of different strategies of treating the same number of individuals but using different strategies to identifying who to treat. And one of the strategies that we looked at was that of actually treating an infected case and their infected primary contacts. So if we picked an infected case, missed it, Tam, you can see where they're supposed to be. It slipped a bit. And we'll move quickly on. So that's the case of the ring treatment. So at each treatment epoch, or a particular point in time, treat a node who's infected at random and then treat all of their primary contacts who are also infected. All of these approaches to treatment are only treating infected nodes. Random treatment is just treat nodes at random across the network. Then there are 2 mechanisms that depend on degree. So of course this is quite hard to realise in practice because you'd need to know the whole contact network in order to implement the strategy, but it's worth thinking about. Do you treat those nodes who have the most contacts? Do you treat those nodes who have the least contacts? The ring treatment we've discussed. And then there are various mixed strategies. Treat a person who's infected and their primary contacts, but only those contacts who've got high degree, and choosing 5 or 3 as the cutoff. A 2-ring is you treat an infected nodes, all their primary contacts, all their secondary contacts, and so on. So what happens? Well, there are, although these lines look quite together, they're quite different from one another. Random, the baseline is known, if you don't treat anyone, is the black line up the top. Completely random choice of treatment is the thick red line. It's not the worst of the methods shown. In fact, the worst of the methods shown is only focusing on the high degree nodes, but really random, only focusing on high degree nodes and only focusing on low degree nodes are the worst of the various strategies tried. What the main point of this diagram is that actually the best strategies are those that do take some account of the network in the treatment strategy. So treating a node and their infected partners is more effective than treating the same number of nodes at random. Interestingly, there's a very large drug trial going on at Melbourne at the moment testing that hypothesis out, and we all wait with bated breath on the outcome. So, to finish up, this class of models, I think, is actually a very broad and flexible framework to think about building models for networks. We've modelled these kinds of networks, but many others besides. We can tell you about credit amongst bankers in 15th century Florence. We can tell you about interactions amongst cattle. We certainly know about trust amongst lawyers, all sorts of things. And surprisingly, that social circuit model is quite useful in just about every case. And the conditional estimation strategy that we've used to estimate a model from these fragments of the network is actually potentially quite exciting, because it means we can actually use network techniques to understand contact patterns and interaction patterns in many more situations, because the collection of network census data is actually quite a hard thing to do often. I mean, there are some cases where you can get it for free. Co-citation networks and all sorts of things like that are much easier to get the whole network, but in many cases that we're interested in, we can't. So this extends the applicability quite well. And as we've shown, these models are potentially useful for these kind of what-if scenarios. There's lots of interesting modelling questions that arise in this context, enough to keep me and many others busy for many years. And I'm particularly excited about thinking about the last one, how to deal with sampling from more dynamic networks. So, in fact, the original data from which this study was drawn was one where data was collected longitudinally, and we're yet to exploit the information we have from the longitudinal component. But with that, let me stop, and of course I'd welcome any questions. Thank you.
Thank you very much, Pip. That was really interesting. We've got time, about 20 minutes for questions. Eddie?
Yeah, thanks. So I've got two questions about error. So first, how big a sample of the network do you need to sample to make these work? And two, invariably networks there'll be error in how to report this. So how do you factor error into? Can you integrate across different types of networks and do a distribution? How do you count that?
Yeah, no, they're both really good questions. First of all, how much of the network do you need to see? The answer to that really does depend upon the structure of the network. If you happen to have a random network in front of you, you actually need a relatively small fragment of it. But if it's a network that's much more complex, so if I go back to my hierarchy of models, the further down this part of the diagram you are, the more data you would need to actually kind of see enough of the network statistics that are sufficient statistics for the model to estimate it. But as a simple rule of thumb, you need to see, enough. Just having a quick look at our model, our data actually, you can see that there's, I think we've done pretty well from really quite a small fragment of the structured part of this network. We've done quite a lot of work, quite a lot of simulation work to actually understand, suppose we know the model, how big a sample do we need to take in order to recover the estimates quite well? Actually, the fragment, it can be quite small, like 5 to 10% is actually not too bad. In relation to error, this is of course a stochastic model, so we expect there to be some error in the data, and we hope that it being a probabilistic model protects us to some degree. But we don't really have a good sense of how a measurement model for the tie variable itself would interact with the probability model for the whole network. that would actually require a more complicated model again to try and unpack that. Yeah. Yeah, well, that's, I mean, in many ways, this is a kind of, and this technique that we've used, we're actually using to get bootstrap estimates for very large networks, because even this is quite computationally intensive. If we've got a network with 10,000 nodes, it's actually going to take, we've had the Victorian cluster tied up for, you know, many weeks doing some of our estimations on big networks. This idea of actually sampling from an observed network and using that to kind of get very close very quickly to the actual parameter estimates is quite a good computational device. Yep.
Can I ask, just ignoring the humanity of my question altogether, and that with the absence of it, have you done any modelling so far as the financial implications of how you treat this kind of modelling scenario? Because clearly a government will be immediately seeing something like this. Okay, so what's the cheapest way we can do this most effectively? Have you done any modelling on that?
We haven't done anything on the numbers, although I have to say that trial that's going on at the moment, it's a simple one in the sense that we're assuming the same cost of delivery of the treatment to every case. But of course, if you're treating a cluster, you might actually be able to treat them more effectively or efficiently or cheaply than if you're treating people completely at random. But so no, the answer is no, but it's kind of interesting question.
Do you think you could extrapolate the injecting network from networks you have more information on, say Facebook or some social media?
Yeah, that's a really good question. I actually started my network life thinking about the interdependencies across networks, and there are definitely quite strong regularities from one network to another. In fact, in this particular case, we have 3 networks. We've got who do you actually share needles with? We have this one, who are you together with when you inject? And we have another one, who are you friends with? And in fact, each of those is just a sparser version of the one that sits above it. And in many cases, we chose the middle one because that was actually most correlated with the biological network that I mentioned before. But I think while it won't substitute for either of the other two, in many cases the logical place to start would have been the who do you actually share a needle with, but we just suspect people were not very honest about answering that. So we have used one network as a proxy for the other, and in the transmission model, because that's probabilistic, you kind of adjust for that a little bit.
And have you been able to confirm through phylogeny of the virus, this network?
Well, to the extent that we've only got the phylogenetic information on these people, and yes, we have been able to confirm it in the sense. It's not perfectly correlated, but it's definitely correlated. And of course, there are some complicated things going on. So some people have got multiple viruses. Yeah. Yeah, no, it's a great question, and indeed there have. In fact, there what became quite a certainly well cited, but also quite widely attacked study by Christakis and Fowler, who used the data from the original Framingham heart study to... They found in the records of that study, which was conducted in the 50s, so it's quite a long... I'm sure all of you know more about this than me. A long time ago. It's still going on, yep. But they were using the historical data. They had in their records information about who to contact if they couldn't find one of the participants in that longitudinal study. And they used that as a network to actually look at potential transmission of characteristics of all kinds, including obesity and all sorts of conditions. And indeed, the reason for the controversy about the study was the methodology they used, but they certainly claimed there was evidence to suppose that there was some kind of social transmission occurring through those networks for those sorts of lifestyle characteristics. Some very mean economists used the same method as they had used to show that something like height was communicated socially, which was probably a bit implausible. So, and in fact, the NIH had a series of conferences to actually, which I was lucky enough to go to kind of think about other methodological approaches you could develop to investigate the same kinds of questions, but without being subject to what seemed to be some flaws in their methods. But I think it is a really interesting kind of issue. Steve?
Yeah, a sort of slightly out there question. Given the well-known biological phenomenon of infections manipulating the behaviour of hosts, can you use network analysis like this to see whether infected people are more likely to participate in sharing of media or
In theory, yes. We haven't in practice, but I think particularly if you think about infections that are more immediately visible, so that people are aware of, you would expect that to affect behaviour more radically. So the transmission process and the network come to be coupled processes, each of which affects the other, and absolutely there are methods for understanding those processes from both perspectives, from the network side on individual behaviour and from individual behaviour onto the network. Tom Snyder's our collaborator in this work, has been the sort of lead in developing those models for co-evolution of things.
Have you tested what in any way considered to see your model in the context of high density populated areas, such as India or Bangladesh?
Yes.
Now, I'm originally from Bangladesh, and there's a serious concern about trafficking, and people are paid to take drugs.
Yes.
And also, technologically they are improving, particularly mobile network, Facebook, etc. We can see a huge number of unidentified and identified cases of HC in the place. So I'm just wondering whether you have thought about it or considering how this model can be tested, or there can be some sort of work.
Yeah. That is also a really great question, because it brings up the cultural factors influencing networks, and they're not to be ignored. We have done a little bit of work in Laos, and we know that the kinds of networks that we see, in fact that was in relation to HIV, we know those kinds of networks are different in structure to the kinds of networks we see relevant to HIV in Australia. So it really does mean we need to actually estimate the model for the context in which we're interested in working. And I think the whole question of how we think about, in a much more sophisticated social theoretical sense, the interplay between culture and network structure, that's very much an open problem, but one that's really interesting.
Can I just check whether there's any questions at the remote sites? Nope. Okay, well, if there's no more questions, can we just thank Pip once again for coming to talk to us today?
Thank you, I enjoyed that.
Very interesting.
Keeps me off the streets.